Our topic for today is on peptide sequencing. So peptide sequencing, similar to nucleotide sequencing, is the process of determining the specific order, the specific sequence of amino acids in a protein. So each peptide is made up of amino acids, as you should know, and they're linked together in a specific sequences through peptide bonds. And this defines their structure and their function as a fully folded, fully mature protein. But understanding this sequence is essential because it reveals insights into the protein's role in the different biological processes of different organisms. Um, Apart from that, the sequence also helps us identify the proteins if we're looking for specific proteins in a sample. And it can also aid in understanding and studying diseases and also in developing medication and developing therapeutics. Okay. So when it comes to the importance or the possible applications of peptide sequencing, most of the current studies around it link genomic information to functional proteins. So genomics kasi, it provides the blueprint, in the form of DNA, while proteomics reveals to us the end products that DNA codes for. So peptide sequencing allows us to connect DNA and RNA sequences to functional proteins. So it bridges genomics and also protein expression. Another set of studies that make use of peptide sequencing would be in terms of gene annotation and verification. So what peptide sequencing does is it validates gene predictions and it helps annotate genomes by allowing us to confirm which genes are actively expressed as proteins. So this is especially useful in genomes that are just newly sequenced, yung hindi pa masyadong alam or identified. Sorry, I'm admitting a lot of your classmates. Peptide sequencing is also useful for evolutionary studies. So when we compare peptide sequences across different species, we can trace evolutionary relationships and identify conserved proteins. And conserved proteins are always of interest because the running hypothesis is that they are the ones that are essential for life, for survival. Peptide sequencing is also important in order for us to understand genetic variants. So peptide sequencing helps us to analyze how genetic variations affect protein structure and function. And this is particularly relevant for studying variants that contribute to diseases. Okay. So let's look in a little bit more detail into the applications of peptide sequencing. Okay. So let's look first at function annotation. So protein sequencing allows researchers to identify proteins based on unique peptide sequences, especially when we're working with complex samples containing multiple proteins. So by comparing sequenced peptides to known protein databases, we can determine which proteins are present. We can determine what version of the protein is present if we know that there are variations. And this is essential for understanding biological functions and interactions. Okay. Next, protein sequencing is also important in creating what we call proteome maps. So this helps to catalog all proteins within a cell, within a tissue, or even within a whole organism. So this is vital in studying how different proteins respond to changes in environment, how different proteins respond to disease states, lalo na kung cancer, kung stage 1, stage 2, stage 3, or stage 4, or even how proteins respond to medication, to therapeutic interventions. So that's another application of peptide sequencing. Next, it's also very useful in terms of quality control. So protein sequencing helps ensure the fidelity, the correctness, the repeatability of recombinant proteins in industrial and research settings. So we have to confirm that engineered products match their intended sequences para sigurado rin natin na na-maintain nila yung desired functionality na gusto natin. They're also important in the field of drug discovery, particularly in target identification and validation. So when we sequence proteins that are associated with a particular disease, we can identify potential drug targets. So for example, proteins that are overexpressed or underexpressed, or proteins that are mutated in certain diseases. All of these proteins can serve as targets for drugs that are designed to inhibit, increase, or just generally modulate their function. So peptide sequencing allows us to identify these variants of interest. Okay. It's also important for antibody and vaccine development. So, kasi protein sequences can reveal epitopes. So, these are specific repeating, specific sequences that indicate a particular variant or a particular functionality. And epitopes are useful kasi sila yung parang ID tag na ginagamit ng immune system natin in order to recognize if a protein or a substance is foreign or local to our biological systems. So, in peptide sequencing, we get to know these epitopes, and so it aids in the development of antibodies and vaccines. And this is crucial for, the ever-increasing number of infectious diseases that we have, and also for creating therapeutic antibodies that target specific protein structures. Okay. Peptide sequencing is also crucial in lead compound optimization. Lead ha, hindi lead, not the heavy metal. Lead compound optimization. So when we do the process of drug discovery, this often involves designing small molecules that bind specifically to protein targets. So when we know the exact sequence and structure of the protein targets, we can actually create models of the interactions with the drug candidates that we already have. So this leads to the development of more effective and selective drugs in a cheaper way. Kasi hindi kailangang lahat i-synthesize. Pwede tayong gumamit ng bioinformatics to predict the interactions that can potentially happen. But again, ang core nun, ang basis nun, would be the peptide sequence. Okay? And so, it's also not surprising that peptide sequencing is very important in order to reduce off-target effects. So, when we have precise protein sequencing data, this allows us to design drugs that specifically interact with disease-related proteins. So, naiiwasan natin yung interactions with other proteins that are not the target of the medicine. So, this improves drug safety profiles, and that's why newer versions of medication usually have less side effects, and sometimes they're a little bit more expensive if I'm sorry about that. If the company or group who made them decided to patent the final or newer version of the therapeutic agent. So even the available medication that we have now is open for improvement the more we learn about the target proteins that they have. Okay? Now, in terms of biomarker research, peptide sequencing is also applied in terms of disease diagnosis and monitoring. So, in protein sequence, we end up identifying specific biomarkers. So, these are proteins that are uniquely expressed or modified in certain diseases. So, for instance, we can now detect cancer-specific protein isoforms or modified proteins. So this can help us diagnose cancers early and also monitor disease progression. For example, sa breast cancer lang, there is a growing number of women who are opting for preventative mastectomy. So when their doctors are already starting to detect protein biomarkers that indicate the possibility of them developing breast cancer, even before they develop tumors, they already opt for mastectomy to avoid the disease. So, may mga ganong cases na tayo because of the development of advanced or early stage diagnostic tools. Okay? Now, in terms of personalized medicine, so nag-i-improve na rin ang ating ang ating applications because of peptide sequencing. Kasi we can get biomarkers from unique protein sequences. So this enables individualized treatment plans based on the unique protein expression that we see in our patients. So this is especially valuable in fields like oncology for cancer, where treatments can be tailored to the specific mutations and protein expressions that are present in a patient's tumor. So yun kasi yung difficulty when it comes to cancer. Even though the progression is similar across all patients, the mutations that you see in the tumors themselves cause some of the cells to behave differently from others. And that's why cancer, among all other diseases, require personalized medicine in order for it to be more effective to the patients that we have. Okay. Next. So peptide sequencing is also useful for creating predictive and prognostic indicators. So it sequencing kasi allows researchers to discover biomarkers that predict disease risk or patient outcomes. So parang yung diagnostic kasi, meron ka ba or wala ka bang sakit? Yung prognostic markers naman is um, magsusurvive ka ba? Ano yung likelihood na magsusurvive ka if you have these markers? So it also helps in managing the kind of treatment if we're aiming for a cure, if it's even possible, or if we're aiming for just management of symptoms. Okay? So, for example, when we're sequencing cardiac-related proteins, this can reveal markers that indicate a higher risk of heart disease or also help forecast recovery outcomes after, let's say, a heart attack or a stroke. So, mas ma-manage mo yung expectations ng patient given the effectiveness of the treatments that we already have. Okay? Peptide sequencing also allows non-invasive diagnostic tools. So may iba kasing diagnostic tools na yung magbabiopsy ka, kailangan mong operahan yung patient even before maging sure ka if may sakit siya or wala. So protein biomarkers that are determined through peptide sequencing can now be used to detect the presence of a disease just using blood, urine, or saliva samples. So these are definitely useful non-invasive diagnostic tools. So by sequencing these proteins, we get to discover potential biomarkers that are easier, cheaper, less painful, less intrusive, and so we make disease detection not just more efficient, pero more appealing. Marami kasing pasyente na natatakot magpunta sa doktor kasi, yun nga, diagnostics pa lang, invasive na, expensive na. But the cheaper we can make it, the faster we can make it, the less painful we can make it, the higher the chance that more people would be open to getting checkups from their doctors. Okay. So now that we've talked about its applications and its importance, ang laki ng impact ng peptide sequencing, ano-ano ba yung technologies na ginagamit in order for us to do peptide sequencing? So when it comes to peptide sequencing, pag nucleotide sequencing, kasi parang ang dami na nating options, no? Especially with NGS. For peptide sequencing, we're actually focusing on main two main techniques, which we'll talk about in detail. But just to give you an overview, the first one is yung Edman degradation. So this is a chemical method where amino acids are sequentially removed, isa-isang tinatanggal, from the N-terminal portion of the protein, and isa-isa silang ina-identify. I guess yun yung version or counterpart ng Sanger sequencing pagdating sa peptide sequencing. And then there's also mass spectrometry, which is now the more widely used, considered more modern technique, where peptides are fragmented and the resulting ionized pieces are analyzed so that we can infer or try to detect the amino acid sequence. So MS or mass spectrometry, particularly yung version na tandem mass spectrometry, is now the most common method because it can handle complex protein mixtures and modified protein sequences with high precision. Whereas, if you compared it dun sa traditional Edman degradation, this one is mainly used for short sequences or yung purified concoction, yung alam mong isang uri na lang ng protein yung meron dun sa solution. So, yun. So, right now, let's focus on how Edman degradation works. So, as I mentioned earlier, this is the classic method of peptide sequencing that sequentially, pa isa-isa, na nagtatanggal, nagre-remove ng amino acids from the starting point of the peptide, which is considered the N-terminus, the nitrogen end of your peptide. So, this is called Edman degradation kasi it was developed by Per Edman in the 1950s. So, this was considered a revolutionary technique in protein chemistry kasi it was the first ever effective method in order to identify peptide sequences. So, to do Edman degradation, you have to chemically tag and selectively remove one amino acid at a time from the N-terminal of a peptide chain. So this happens in a cyclical manner. After each cycle, the released amino acid derivative is identified, and this allows us to deduce the sequence one residue at a time. So yun nga, dahil doon highly accurate siya, but it's also a little slow. So while it's historically the primary method for protein sequencing, Edman degradation is now limited to short peptides and specific applications because mass spectrometry has ended up being more efficient and also cost-effective. So we will be now discussing the different steps in Edman degradation. So the first step, so it begins by treating the peptide with a chemical called phenyl isothiocyanate or PITC. And you have to do this under alkaline conditions. So the pH, as you can see from the illustration, is 8. So medyo alkaline dapat. So what happens is that PITC reacts with the free amino group of the N-terminal amino acid to form a derivative called phenylthiocarbamyl or PTC. Okay, so yun yung derivative na nakikreate. From there, we move forward to the second step. So the peptide with the PTC derivative is treated with anhydrous acid, which causes the cleavage of the N-terminal amino acid as a phenylthiohydantoin or a PTH derivative. And so natatanggal na yung isang part, nagiging PTH na siya. So this leaves the rest of the peptide intact. Now, this step generates a modified amino acid, which can be identified using chromatography or other analytical methods. So, paano na-identify yun? No? The released PTH amino acid derivative is identified by comparing it to known standards using chromatographic techniques. So typically, we use HPLC or high-performance liquid chromatography. So each amino acid derivative has a unique retention time, how long it stays on the chromatogram. The sequence can be determined by analyzing the order of PTH amino acids released. Like, ano yung pinakaunang gumalaw, ano yung susunod, gaano sila katagal gumagalaw, and so on. Now, the remaining peptide, that's now shorter by one amino acid, undergoes the same process again, with PITC reacting with the new N-terminal amino acid. So this cycle is repeated for as many residues as necessary to determine the peptide sequence. Now, Edman degradation is usually effective for proteins that are about 30 to 50 amino acids in length. So yun yung ibig sabihin natin when we say short protein sequences. If it's any longer than that, then Edman degradation is not really advisable. But I'm going ahead of myself. Apart from length restriction, ano-ano pa ba ang limitations ng Edman degradation? So as I mentioned earlier, Edman degradation is generally effective for peptides that are just about 30 to 50 amino acids long. And if you have larger proteins, they have to be enzymatically or chemically cleaved into smaller peptides before you can sequence it using this technique. So this makes Edman degradation impractical for sequencing large proteins, which are now typically sequenced using mass spectrometry. Now, another limitation of Edman degradation is the requirement for a free N-terminus. So if the N-terminus is blocked because of some modification that happens, like acetylation or formylation, it must be chemically modified or otherwise unblocked before sequencing can be done. So this is particularly limiting when you're dealing with modified or processed proteins, which, is what happens to a lot of biological proteins that undergo N-terminal modifications in order for them to become functional. So maraming hindi nasi-sequence dahil dito in the past. Okay? Next, there are also limitations when it comes to sensitivity. Edman degradation is less sensitive than modern techniques, and it actually requires a relatively high quantity of pure peptide. So high concentration na, pure pa siya. So when we say high concentration, the proteins have to be present in micrograms. Pero most of the time, sobrang trace amounts lang yung makikita natin sa solution. So yung mga yun, hindi na natin na-detect through Edman degradation. And this is because the degradation efficiency decreases with each cycle. So it limits the usefulness of the technique for very small samples or peptides with low concentration. Next, as you can predict from the process, it's very slow and labor-intensive. Each cycle of Edman degradation is time-consuming, particularly if you're dealing with sequences na nasa 40 to 50 amino acids na. Kasi nga, it's performed one amino acid at a time. Also, it's a manual process. Hindi siya katulad ng PCR na automated na siya. So, at each cycle, you have to be actively involved, transferring solutions and so on and so forth. So you can imagine how labor intensive it is and how it requires considerable expertise from those who perform it. So it's definitely a slower and less efficient method compared to automated methods. And then lastly, it cannot detect post-translational modifications or PTMs. So it gives us the primary sequence of amino acids, but if the protein has been phosphorylated, glycosylated, or methylated, hindi na natin yun malalaman. Kasi nga, amino acid lang yung kaya niyang i-detect. So the issue there is that post-translational modifications play critical roles in terms of protein function and regulation. So this makes the method less informative when we're trying to fully understand modified proteins. Okay? So buti na lang meron ng mass spectrometry. So this is a very powerful analytical technique that's widely used in modern biochemistry and proteomics when you're trying to identify the sequence of peptides and proteins. So this has replaced traditional methods because of its speed, because of its sensitivity, and because of its ability to analyze complex mixtures. So paano ba nag-work ang mass spectrometry? So it works by ionizing molecules and then measuring their mass to charge ratio. So when we say mass to charge, that's M/Z ratio. So this is determined by allowing the molecules to travel through an electric or magnetic field, and you determine how fast they get to travel. So pag ganito yung charge, pag ganito sila kabilis, ito yung molecule na yun. So yun yung mass to charge ratio. So in terms of the steps that are involved, the first one is ionization. So the sample is first ionized, meaning it's given a charge so that it can be manipulated using electric or magnetic fields. There are different ionization techniques, but there's two that's more commonly used. If you've watched the Independent Learning Week video, you might have heard me mention both. The first one would be electrospray ionization. So this produces charged droplets that evaporate, leaving charged analyte ions. So electrospray ionization, or ESI, is ideal for large biomolecules like proteins and peptides. And the good thing about it is it works at atmospheric pressure. So you don't really have to control pressure too much. The other option is MALDI, MALDI, or matrix-assisted laser desorption ionization. So in this method, a laser pulse vaporizes and ionizes the sample that's embedded in a matrix. So MALDI is popular for analyzing complex mixtures and samples on a solid matrix. Now, after ionization, the next step would be a mass analyzer. So the sample ions are detected through a mass analyzer, which separates ions based on their their mass to charge ratio. So common types of mass analyzers, we have the TOF analyzer, or the time of flight analyzer. So this measures the time it takes for ions to travel a known distance. So basically, follows the principle that the lighter your ions are, the faster that they travel, while heavier ions are slower. But there's also a quadrupole analyzer. So this uses oscillating electric fields to filter ions based on their mass to charge ratio. And then there's also an orbit trap, I think. Yeah, an orbit trap analyzer, which follows the principle na ions basically orbit in an electrostatic field. So their frequencies relate to their mass to charge ratio. And this provides so far the highest resolution data. Pero I've never encountered a mass spec that uses an orbit trap. Most of the mass spec, I think in the Philippines, still uses time of flight, which I think is still very relevant pa naman, no? Anyway, the next step would be the detector. So the separated ions would reach the detector stage where their arrival is recorded as signals. So the intensity of each signal corresponds to the abundance of ions at a specific mass to charge ratio. So the resulting mass spectrum shows peaks for each mass to charge ratio, allowing identification of ionized molecules based on their masses. Okay. I think in the lab, we've prepared some data sets for mass spectrometry for you to analyze, for you to determine what amino acids are present. So yeah, we'll be doing some of that in the next set of activities that we'll have. Okay. Now, when it comes to tandem mass spectrometry, yun yung MSMS, although hindi na siya ganun ka-commonly used ngayon, it's a specialized form of mass spectrometry that's used to sequence peptides and identify proteins by analyzing their fragment ions. So MSMS involves two stages of mass spectrometry with the fragmentation step in between. So let's go through the different stages of MSMS. So first stage first, in MS1, the sample's peptide ions are separated based on their mass to charge ratios. So each peptide ion corresponds to a fragment of a protein and is selected based on its mass to charge ratio. So a specific peptide ion is then isolated. So this is known as the precursor ion for further fragmentation. So the next stage is fragmentation. So the precursor ion is now fragmented using one of several methods. So the three most commonly used fragmentation method would be collision-induced dissociation, or CID. So this one. So the ion collides with an inert gas, for example, nitrogen, and this causes it to break at specific points along the peptide backbone. Another method would be higher energy collisional dissociation, or HCD. So similar to CID, this one generates higher energy collisions though. So it still facilitates a collision, but using higher speeds and higher energies. So it provides a broader range of fragment ions. And then lastly, there's ETD, or electron transfer dissociation, wherein electrons are transferred to the precursor ion, causing it to fragment differently. So this offers, or this often preserves post-translational modifications. So if you're interested in post-translational modifications, you often use ETD. So the fragmentation now produces a set of ion fragments. Usually they're called B ions and Y ions. So we'll talk about them in a little bit, which represent portions of the peptide with information about the amino acid sequence. Okay. Next. So second stage, MS2. In MS2, the fragment ions are separated and detected, producing a spectrum that shows the mass to charge ratios of the fragments. So by analyzing the patterns of these ions, we can deduce the amino acid sequence of the peptide, while the difference in the mass to charge ratio between successive fragments often corresponds to the mass of a specific amino acid, revealing the order of residues in the peptide. So this is a table that summarizes the differences between Edman degradation and mass spectrometry in terms of mechanism, peptide length, sample requirement, speed, sensitivity, ability to detect post-translational modifications, applications, and limitations. It's an easier way to study for the exam. So you can take a photo of the slide, very, very useful. Um, but generally, Edman degradation has a, I guess, precise, stepwise approach, making it useful for short peptides or small scale studies. But it is limited by the need for purified samples and an unmodified N terminus. On the other hand, mass spectrometry, particularly with MSMS, it's become the preferred method for peptide and protein sequences in a lot of the labs that we have today because it's versatile, it's able to handle complex and low abundance samples, and it has the capacity to identify post-translational modifications, which are critical for functional studies. Okay, so now let's put the focus on mass spectrometry-based peptide sequencing. So we will be looking at mass spectrometry in a little bit more detail. So we'll go through some of the steps that we've already talked about earlier, pero mas malalim ng konti. Okay? So let's start first with ionization. So as we talked about earlier, ionization is one of the crucial steps in doing mass spectrometry because it enables the analysis of peptides by converting them from neutral molecules into charged ions. So this is essential because of the following 5 reasons. The first is it allows mass to charge ratio analysis. So most mass spectrometers separate ions based on their mass to charge ratios. So without ionization, peptides would remain neutral and hindi na natin sila mamamanipulate using electric or magnetic fields in the mass spectrometer. So making it impossible to measure their mass to charge and determine their masses. The second importance of ionization is the ability of ionization to generate multiple charge states for larger biomolecules. So for larger peptides and proteins, multiple charges can be given or imparted to them through ionization methods like yung ESI, so electrospray ionization. So this is essential because large molecules, if singly charged lang sila, if isang charge lang yung nabigay, they would have very high mass to charge ratios that may exceed the mass range of the mass spectrometer. So multiple charging reduces their effective mass to charge ratio, allowing the mass spectrometer to detect and analyze larger peptides and proteins. Okay. The third is its ability to improve detection sensitivity and signal quality. So efficient ionization enhances the sensitivity of mass spectrometers kasi more ions lead to stronger signals. So this enables the detection of low abundance proteins. For instance, if you use ESI, yung electrospray ionization, or MALDI, again, matrix-assisted laser desorption ionization, these are both highly sensitive ionization techniques, providing strong signals even from very, very, very small protein samples. Now, high-quality ionization also improves reproducibility and reliability of the data, which is crucial for precise peptide sequencing and protein identification. The fourth is, yun nga, ionization allows for MSMS. Ionized peptides are important, if not the core requirement for fragmentation in MSMS. So this is where specific bonds along the peptide backbone are broken to generate fragment ions that reveal the sequence of amino acids. So fragmentation techniques such as collision-induced dissociation, yung minention natin kanina, rely on ions to produce consistent fragmentation patterns that are essential for deducing the peptide's sequence. And then lastly, ionization allows us to identify post-translational modifications. So again, when a protein has post-translational modifications, this can add or change the mass of our peptides. So since each modification, whether it's phosphorylation or glycosylation, changes the peptide's mass, the resulting ionized peptide displays a distinct mass-to-charge ratio that can be identified and then analyzed. So this enables researchers to determine not only the peptide sequence, but also important functional modifications that affect protein behavior. So let's now put a focus on MALDI and go through some of the considerations when we're deciding to use this technique. So in terms of sample preparation in MALDI, the analyte, which is your protein solution, is mixed with a matrix. So this is a small organic molecule that absorbs laser energy efficiently. And then once it's already mixed with the matrix, it's deposited on a metal plate. And then the matrix helps absorb energy from the laser, protecting the sample from full degradation. Now, when laser ionization is finally being done, a pulsed laser beam, so we often use a UV laser, it irradiates the sample matrix mixture, and the matrix absorbs the laser energy, causing it to vaporize and carry the analyte into the gas phase. Now, the next stage would be desorption and ionization. So the laser energy causes both desorption, which is the release of analyte molecules into the gas phase, and ionization. So this typically generates singly charged ions for the analyte molecules. Now, MALDI usually produces ions with a single positive charge, making it ideal for generating clean, simple mass spectra data. So the advantages of MALDI is that it's good for large molecules, including peptides, proteins, and polymers, because it produces singly charged ions, resulting in a simplified spectrum with a high mass-to-charge range. It's also considered high throughput because, samples can be prepared in an array format. It makes it efficient for large-scale proteomics studies. And finally, since MALDI is considered a soft ionization technique, it imparts minimal energy to the sample and produces fewer fragment ions. Therefore, it preserves the integrity of larger biomolecules for accurate mass analysis. So MALDI is commonly used in proteomics for protein identification, for large biomolecule analysis, and the analysis of polymers and other macromolecules. It's also effective for tissue imaging and other spatially resolved analyses. On the other hand, we have electrospray ionization. So when it comes to sample preparation, the analyte is dissolved in a solvent. So this is often a mixture of water and an organic solvent like methanol or acetonitrile. And then it's pumped through a narrow charged capillary. From there, a formation of charged droplets is created by applying a high voltage to the tip of the capillary. So this creates a strong electric field that generates a fine mist of charged droplets. So these droplets carry the analyte molecules and are released as a spray. And then the solvent would evaporate kasi as the droplets travel through a desolvation chamber or heated gas, the solvent would heat up and then evaporate, and the droplets get progressively smaller. So this process continues until only the charged analyte ions are left inside the chamber. Pag nangyari na yun, nagkakaroon na tayo ng multiply charged ions. So ESI typically produces what we call multiply, hindi multiple ha, multiply talaga. Multiply charged ions, meaning that large molecules can have several charges. And as we mentioned earlier, this reduces their mass to charge ratio to a range that is more suitable for mass spectrometers with limited mass ranges. So this multiple charging is particularly useful for analyzing large proteins and peptides. So the advantages of ESI, it can directly analyze complex mixtures, making it compatible with liquid chromatography mass spectrometry or LCMS, where it ionizes analytes directly from the liquid phase. ESI also provides high sensitivity, which is ideal for analyzing low abundance proteins and peptides. But ESI also produces multiply charged ions, which are useful for MSMS, where the peptide is fragmented for sequence analysis. So ESI is widely used in proteomics, metabolomics, and drug discovery when we're trying to analyze complex mixtures, peptide sequencing, protein characterization, and quantification. So I've also created a table that compares MALDI and ESI based on ionization method, ion charge state, analyte type, fragmentation, throughput, and sensitivity. So you can also screen cap this to help you with your review later on. But generally speaking, both MALDI and ESI play critical roles in proteomics and other life sciences. So they have different strengths for ionizing biomolecules. So in the case of MALDI, its strengths lie in its capacity to analyze large biomolecules in a high throughput manner, making it ideal for protein profiling and imaging. ESI, on the other hand, is very useful in coupling with liquid chromatography, in having high sensitivity, and also in its capability to analyze complex mixtures. So the choice between MALDI and ESI depends on the specific application, on your sample format, and the nature of the molecules under study. Okay. Next, we'll be focusing this time on understanding fragmentation. So when it comes to fragmentation, it's critical for identifying and sequencing molecules, especially peptides and proteins. So by breaking down an ionized molecule into smaller, predictable fragment ions, it allows us the possibility of getting detailed structural information. It helps us reveal the composition, sequence, and chemical modifications of the molecule. So these are the 5 reasons why fragmentation is essential. So first, in terms of peptide and protein sequencing, in MSMS, fragmentation helps us to sequentially break peptide bonds, producing fragment ions that correspond to specific parts of the amino acid sequence. So by analyzing these fragment ions, it would then become possible to determine the exact order of amino acids in a peptide. And then fragmentation also allows the detection of post-translational modifications like phosphorylation or glycosylation. So modified amino acids would produce distinct fragment ions that can then be identified. And so this helps us understand more protein function and regulation. Now, fragmentation also helps us understand the structure of a protein. So for small molecules, fragmentation provides structural clues that help us understand its molecular structure. So each fragment ion represents a specific part of the molecule, and by piecing this information together, we can determine the overall structure. But fragmentation can also reveal specific functional groups within a molecule. For example, loss of water or ammonia during fragmentation indicates the presence of hydroxyl or amino groups respectively. So nalalaman lang natin yan through fragmentation. Third is related to identification and database matching. So fragmentation produces a unique pattern or spectrum for each molecule. So in proteomics, this spectrum is then compared to a database of known spectra to identify the peptide or protein. If we don't have fragmentation data, matching would be limited to molecular weight alone, which is often not unique. Maraming proteins and amino acids na magkakapareho ang size. Fragmentation can also differentiate between molecules with identical masses, so these are what we call isomers, by breaking bonds in a way that reveals their structural differences. Then fourth, it helps us determine the amount of complex samples. So fragment ions that are specific to a target compound are used to quantify it within complex samples, meaning samples na may multiple compounds inside it. So this is especially useful in metabolomics or pharmacokinetics, where samples are never pure, meaning they're always made up of a mix of similar compounds. And then finally, fragmentation is important for validation and confirmation of molecular identity. So fragmentation provides multiple data points to confirm the structure of a molecule. For instance, if a suspected structure is proposed, comparing predicted fragment ions with experimental data validates its identity. So let's go through the different fragmentation techniques that we had an overview of earlier. So first, CID. So again, CID involves the collision of an ionized molecule. So this is called the parent ion with an inert gas molecule. So I've already mentioned nitrogen earlier, but an alternative would also be argon. So this all happens in a structure in a chamber that's called the collision cell. So the energy from these collisions causes the parent ion to lose energy, resulting in fragmentation. From there, magkakaroon ng energy transfer. So when a parent ion collides with a neutral gas molecule, kinetic energy is transferred to the parent ion, promoting the breaking of specific bonds. So in proteins, ang nagbe-break peptide bonds. And this leads to the formation of fragment ions. So yun na yung third part, formation of fragments. So the fragmentation pattern is often influenced by the structure of the parent ion. It's also influenced by the types of bonds that are present, lalo na kung may post-translational modification, and the energy of the collisions. So CID generally produces a series of fragment ions that correspond to the loss of specific residues or sections of the molecule, revealing information about the sequence. And then the last part would be detection. So the resulting fragment ions are analyzed using a mass spectrometer, producing a spectrum that displays the mass to charge ratios of the fragments, which can be interpreted to deduce the sequence of the parent ion. So the advantages of CID, first, it's considered a relatively soft ionization technique, meaning it induces less extensive fragmentation compared to other methods, allowing for better retention of the original molecular structure. CID also produces a variety of fragment ions that provide rich structural information, so making it valuable for peptide sequencing. Kasi alam natin yung structure. Now, when you compare it to HCD, so again, HCD is similar to CID, but it operates at a higher energy level. So in HCD, the ionized molecule collides with a neutral gas molecule in a higher energy environment, leading to more extensive fragmentation. So in terms of mechanism, the parent ion is accelerated through a potential difference before entering the collision cell. So pinapabilis na yung movement niya even before pumasok sa collision cell. So this increases its kinetic energy significantly. And when this high energy ion collides with a neutral gas, it has a greater chance of breaking multiple bonds, resulting in a more extensive fragmentation pattern. So yung fragmentation products niya are slightly different from CID. So the higher energy input in HCD typically produces more fragment ions, especially when compared to CID. So this This extensive fragmentation can yield more information about the structure and the sequence of the parent ion. Now, in terms of detection, similar to CID, the resulting fragments are detected by mass spectrometer, and again, the spectrum that comes out is used to determine the structure of your parent ion. Now, the advantages of HCD is that the higher energy levels lead to more enhanced fragmentation, which can provide a more detailed sequence information, especially when you're dealing with larger and more complex peptides. Then HCD is particularly well suited for high-resolution mass spectrometers, such as Orbitrap or your Fourier transform ion cyclotron resonance, or FTICR-MS, which allows for high-resolution analysis of fragment ions. So from there, how do you interpret mass spectrometry data? So particularly in the context of peptide sequencing. So this often involves analyzing specific types of fragment ions known as B ions and Y ions, which I mentioned earlier. So these ions are generated during the fragmentation of peptide molecules through methods like CID or HCD. So understanding the principles behind these ions is important for deciphering the sequence of the original peptide. So how do we differentiate B ions from Y ions? So when a peptide is ionized and then fragmented, it typically breaks the peptide bonds that link amino acids together. So the resulting fragment ions can then be categorized based on their structure. So when it comes to B ions, these ions are generated from the N-terminal, the amino end of the peptide, and are defined as the fragment ions formed by the cleavage of the peptide bond, resulting in a positively charged ion that includes the first amino acid and continues to the C-terminal or the carboxyl end of the peptide. The Y ions naman are formed from the C-terminal end of the peptide sa kabilang dulo. Y ions result from the same cleavage of the peptide bond, but they include the amino acids from the C-terminal end back to the N-terminal end. So parang magkabaliktad lang sila. No? So let's look at how B ion formation happens. So when a peptide undergoes fragmentation, so the cleavage occurs at the peptide bond, generating a B ion. For example, if your peptide sequence is ABC, the B ion corresponding to the sequence AB would retain the N-terminal portion of the peptide. So the B ion is characterized by the mass-to-charge ratio that corresponds to the sum of the masses of the amino acids included in the ion plus a proton. So kasi yung ions na to ay often protonated. On the other hand, for Y ions, so let's say you have a peptide ABC, a Y ion would represent the fragment BC after fragmentation. So this ion retains the C-terminal portion of the peptide. So the Y ion is defined by its mass-to-charge ratio as well, which corresponds to the mass of the amino acids from the C-terminal to the point of the cleavage plus a proton as well. So pareho naman silang may proton. Magkabilang dulo lang nung amino acid. So how do we interpret what they mean? So when you're interpreting mass spectra for peptide sequencing, the presence of both B ions and Y ions are important. So first, when you're trying to identify peptide sequence, the pattern of B ions and Y ions helps reconstruct the original sequence of the peptide. So when you identify the mass-to-charge values of the B ions and the Y ions in the spectrum, researchers can deduce the order of amino acids in the peptide. For example, if the spectrum shows B ions and Y ions, the sequential increase in mass of one and the sequential decrease in mass in the other can indicate how many amino acids are present in their specific arrangement. So para siyang Sudoku na titingnan mo yung pattern ng B, tapos i-compare mo siya sa pattern ng Y para ma-confirm mo kung ano ba talaga yung order ng amino acids. Now, apart from that, they're also very useful in determining charge states. So the interpretation of B ions and Y ions can show us the presence of multiple charged states pag nagkakaroon ng overlapping peaks sa spectrum. So careful consideration of the charge states is essential to accurately identify kung ano ba talaga yung tamang sequence ng B ions and Y ions. But it also provides us a hint on possible modifications that are existing in the proteins. Then finally, B ions and Y ions help support database searches. Kasi pag tinitignan nyo yung peptide sequence databases, they both contain yung identified B and Y ions. So the information can be used to search protein databases for matches. So it helps us identify the protein from which the peptide originated from. So, ano ba ang bioinformatics na ginagamit natin when we're dealing with peptide sequencing? So there are two primary approaches that are utilized to identify peptides and their corresponding proteins. First, database sequencing. Second is de novo sequencing. So each method has distinct applications and strengths, making them suitable for different scenarios in proteomics. So let's look first at database searching. So you're probably familiar with this already since database searching is also largely done in genomics. But yeah, database searching involves comparing the experimentally derived peptide sequences to a pre-existing database of known protein sequences. So the goal is to find matches based on observed mass-to-charge ratios of the peptide fragments. So it has its own set of advanced algorithms. So commonly used would be Mascot, Sequest, or Andromeda. I think I'll go through some of this later. Sana umabot sa time. But these are used to score potential matches based on the similarity of the observed fragment ion patterns to theoretical spectra generated from database sequences. So these algorithms evaluate factors like the number of matching fragments, mass tolerance, and the overall statistical significance of the match. So ang applications when you're doing database searching is, of course, to determine what protein you have. So it's effective, of course, only when proteins are well characterized and represented in the database. But it can also be used to identify post-translational modifications, as long as, again, the post-translational modifications are known. So the strengths of database searching is that it's generally faster as it relates your data to pre-existing data. So it allows for rapid identification of proteins in large data sets. And the use of statistical methods to evaluate the confidence of matches provides a level of rigor in the identification process. However, as you might have predicted from the previous slide, the effectiveness of database searching is contingent upon the completeness and accuracy of your database. So if a protein of interest is not included, you won't be able to identify it. And so this approach cannot identify novel proteins or sequences that have not been previously cataloged. In terms of de novo sequencing, this constructs the peptide sequence directly from the mass spectrometry data without reference to any existing database. So you rely on the analysis of the observed fragment ions to deduce the amino acid sequence. So the method involves interpreting the fragmentation patterns of the detected peptides to piece together the sequence based on known chemical properties of the amino acids and typical fragmentation behavior. So in terms of applications, de novo sequencing is particularly useful pag hindi na useful yung database. Kasi you can now identify novel peptides or proteins that are not present in previous publications or databases. And this method can also help identify unexpected modifications or novel variants that again may not be represented in existing databases. So since it doesn't rely on pre-existing sequences, de novo sequencing can identify completely new peptides or variants, and it provides detailed information about peptide sequence, including potential modifications. However, de novo sequencing is computationally intensive and often requires sophisticated algorithms of your design, and it can require software for accurate sequencing assembly. So this approach may be less efficient if you're working with large data sets compared to database searching, as it involves more complex analyses of each spectrum. Okay. But of course, you are not alone when you're dealing with proteomics. There are common bioinformatics tools that you can use. So the most popular one is Mascot. It's a widely used search engine for peptide identification from mass spectrometry data. So it was developed by Matrix Science and is known for its efficiency in comparing experimental data against protein databases. So it has features, has the following features. Mascot compares spectrometry data to protein databases, allowing users to identify peptides by matching observed peptide mass fingerprints or fragmentation patterns to theoretical spectra. It also has a scoring system to assess the quality of matches based on factors such as the number of matching fragments, mass accuracy, and statistical significance. Mascot also supports the identification of modified peptides, yung may post-translational modifications, by allowing users to specify known post-translational modifications during the search. And then it offers a very user-friendly web interface for data submission and analysis, making it accessible for researchers without extensive bioinformatics training. So in terms of application, Mascot is primarily used for identifying proteins in complex biological samples, such as those derived from proteomic studies, and it is effective for analyzing samples with complex mixtures so that we can determine protein composition. Now, an alternative, which is the one I used when I was in the University of Washington, is Sequest. But Sequest is now known as Comet. Nagpalit na ng name kasi nag-iba na ng funding agency. But it's another algorithm that's developed by the University of Washington for identifying peptides for MSMS. So it focuses on the analysis of fragmentation spectra to deduce peptide sequences. So in terms of feature, Comet performs de novo sequencing by analyzing fragmentation patterns of your B ions and Y ions to identify peptides based on their mass spectra. But similar to Mascot, Sequest also compares experimental protein spectra against protein databases, but it uses a slightly different algorithmic approach to score matches. And then it provides multiple scoring metrics, allowing users to evaluate confidence of peptide identification based on the quality of matches. So in terms of applications, Sequest is particularly well suited for analyzing data from MSMS experiments, making it valuable for peptide sequencing and protein identification. And it can also identify and characterize modified peptides and variants based on fragmentation patterns. And then I think this is the last one for bioinformatics tools. There's MaxQuant. So it's a software platform designed for quantitative proteomics and peptide identification. So people usually use this when they want high throughput proteomics data. So features, MaxQuant provides advanced features for label-free quantification, allowing us to compare protein abundances across different samples. It also utilizes a modified version of the Andromeda search engine for peptide identification, combining both MS and MSMS data to enhance accuracy. Um, MaxQuant also offers integrated analysis tools for post-translational modifications, protein group identification, and statistical analysis of the results. And it's been described as having an easy to understand graphical user interface, making it accessible for users with varying levels of bioinformatics experience. So in terms of applications, MaxQuant is commonly used in large-scale proteomic studies where multiple samples are analyzed, allowing for, I think, more accurate quantification and identification of proteins. And it's very useful in identifying and quantifying post-translational modifications. So it's very useful if you want to do a detailed study on the functional regulation of proteins. But of course, peptide sequencing has not yet been perfected. There are certain issues with peptide sequencing, including the complexity of post-translational modifications. So PTMs are, including phosphorylation, glycosylation, and ubiquitination, can introduce significant complexity into peptide sequences. So the presence of these modifications can alter the mass and fragmentation patterns. So it complicates identification and quantification. Um, PTMs can also occur in multiple sites within a protein. So this can lead to a range of isoforms. So this variability can make it difficult to determine the specific modification present in a given peptide. And then finally, in terms of challenges, some PTMs are present in low abundance and may be masked by more abundant non-modified peptides during analysis. So this leads to underreporting or missed identification. So our current limitations in terms of technology is that many database search algorithms may not adequately account for all possible PTMs, which can lead to incomplete identification. So if a PTM is not specified in the search parameters, it may go undetected. And then quantifying modified peptides accurately can be challenging due to differences in ionization efficiency compared to their unmodified counterparts, potentially leading to biased results. Now, another source of complication would be having, as the term suggests, complex mixtures. So in complex biological samples, multiple peptides may co-elute, leading to overlapping mass spectra. So this overlap can actually hinder the identification of individual peptides and complicate the interpretation of data. So sa halip na makapag-MS ka, ipopurify mo tuloy muna yung sample bago mo siya ma-identify. And then biological samples also often exhibit a wide dynamic range of protein concentrations, with some proteins present at high levels, while others are only present in trace amounts. So high abundance proteins can mask the detection of low abundance peptides. Kaya meron talagang aficionados pa rin of purification before quantification. Now, in terms of technological limitations, so the ability to accurately distinguish between closely related peptides or ions depends on the resolution of the mass spectrometer that you have. So low resolution instruments may struggle to separate and identify peptides accurately in complex mixtures. And then finally, analyzing complex mixtures often requires high resolution instruments, which are more expensive. So, and also they can be time consuming. So sample preparation steps may add further complexity and variability to the analysis. And then last na talaga, when you have very long or highly modified peptides, we might have difficulty dealing with them also. So longer peptides may not fragment predictably during mass spectrometry. So this complicates sequence determination. The complexity of fragmentation patterns can make it difficult to deduce the original sequence from the observed data. So kaya important yung mga databases. And then in highly modified peptides, certain modifications may affect the ionization efficiency during mass spectrometry. So this can lead to poor detection of modified peptides, particularly in the presence of competing ions from more abundant samples or more abundant proteins. Now, technological limitations. Long peptides may exceed the typical lengths used in databases, complicating the search process. So if the database lacks entries for long or highly modified peptides, identification becomes problematic. Tapos, yung de novo sequencing algorithms naman may struggle with very long peptides due to increased complexity and potential for ambiguous interpretations. So this leads to incomplete or erroneous identification. So out of the three complexities that we have, this is the one that we are mostly limited with kasi hindi natin sure kung tama or accurate ba yung interpretation. But anyway, for Thursday, we are scheduled to prepare for a group presentation on the topic. So you will be asked to start the development of a 10-minute presentation on the latest advancements in peptide sequencing. So I'll be giving you your specific group assignment this coming Thursday. And your presentation should highlight recent developments and innovations in the field, demonstrating their significance and potential impact on our understanding of human genetics. So just like in a previous presentation, I need you to use peer-reviewed journal articles published within the last 5 years. And I want you to focus on advancements in human genomics that do not pertain to disease, but rather to aspects such as evolution, adaptation, or non-medical applications. So similar recommendation for presentation structure. You can start with an overview of peptide sequencing and its relevance, and then describe the specific advancement or discovery, include details about the technology or method used and the main findings. And then in terms of significance, explain why the advancement is important, discuss its implications and how it contributes to the broader understanding of human genetics. Include relevant examples or case studies from the journal articles, and summarize the key points and highlight future directions or applications. So since this is a report, you'll need a PowerPoint or similar visual aid to support your presentation. I need your slides to be clear, concise, and visually engaging, and make sure that you stay within the 10-minute time limit. Okay? So the presentation date for this one is on November 11th. So that's next week, Monday. It's going to be online. It's going to be synchronous. But the group can decide whether you want to present live or you want to just play a recorded version of your presentation. Okay? But anyway, as I mentioned, you will be given this Thursday to discuss your topic with your group mates. But I'm also hoping that on the second half of our meeting on Thursday, we can use it, we can use it to start the collaboration for your final project in the lab, which is the expert talk. So I hope that representatives from both lab classes can take charge and have discussions on committees, which members of the class will be part of which committee, finalize your speakers, and so on. Okay? So anyway, more or less that's it for today. If you have any questions or points of clarification, now would be the best time to ask. So if you have any questions, you can type it in the chat or you can also unmute and ask. Okay? Kung wala po tayong questions, kung wala pa tayong maisip, that's perfectly fine. Yes, Yanika, go ahead.
Hi, Doc. Sorry. This is regards to the lab event that we need to plan. If ever we uh don't follow the exact deadline set, like for example, I believe by tomorrow we're supposed to have the list of speakers, I think. I'm not sure. But we're only gonna be meeting po today kasi. So is it okay if we don't hit every mark?
I think yung deadlines that we've provided are just some basic guidelines kasi
Ah, okay.
technically ang deadline nyo is the actual presentation date.
I see.
But we've been given feedback before na if we can give advice na on what steps need to be accomplished and the recommended dates by when the students should accomplish them. Kasi I've heard that some students function better if like alam nila kung kailan yung deadline kesa yung bukas na pala siya, wala pa kaming nagagawa. So we created that, pero we won't really be asking you to submit anything. You can expect possibly me or Doc Ziba to ask you, kamusta na? Meron na ba? Pero yun, we're just hoping that you can keep track of your activities using that outline. But at the end of the day, we will be grading you based on the final event and also the reflection
Okay
paper.
po.
Mm,
Okay po. Thank you so much.
sure. We also have a question in the chat. How many items is the exam? I think it's 85 points. So just 10 points longer than the previous exam. I don't really want to give it too long because we only have an hour and a half. So yeah, it's I think 85 points. For my lab students, I think they're asking if you still need to submit the outline of the elevator speech. You don't need to. We've canceled that output. You can just go straight to submitting your final output. Okay, any other questions? Okay, if you can't think of anything, you can also message me via Animo Space. You can also ask on Thursday. So looking forward to seeing everyone face to face again on Thursday. So thank you so much everyone for attending today's session. Looking forward to seeing you on Thursday. Bye-bye. Thank you very much.